for now, I'm going to be talking a little bit initially about the Message Project, which is a policy initiative to improve how all biomedical health and care research in the UK accounts for sex and gender. This includes cell, animal and human research. So we're looking at a really wide scope here. And what we're focused on changing is practices for data collection, and that's specific to who is included in research, or in case of animals, which animals, as well as ensuring appropriate sex and gender desegregation when data are analysed and reported. Accounting for sex and gender, just to give you some caveats, it doesn't mean powering every study to generate statistically significant findings for every sex and gender group. And it also doesn't mean controlling for sex and gender, which actually renders really important differences invisible in your study data. What it does mean is critically considering the possible impact of sex and gender dimensions throughout the research cycle, designing studies in such a way that possible sex and gender differences may emerge in study data, and reporting transparently on the sex and gender dimensions of study participants or subjects in your research outputs and in your papers. Just a note on definitions, although the terms are quite often conflated, sex and gender are actually terms which have quite distinct meanings, and that distinction is really important in the context of health research. So when I'm talking about sex, I'm referring to biological attributes such as hormones, anatomy, and genes. And when I'm talking about gender, I'm referring to aspects of a person's identity which are shaped by social forces, and that's only relevant for human research. So the question is, why is this project needed? Well, sex and gender, as you probably can expect, play a really key role in determining our experiences of health and of illness. And for example, they shape the medical conditions we develop, the symptoms we experience, the kinds of treatments which are effective, the quality of care we receive, really importantly, disease progression and overall outcomes. So there's lots of ways that they're having an impact here. And yet historically, very little attention has actually been paid to these variables in research. This is translated in many ways into an overrepresentation of male research participants. For example, up to 5.5 times more male than female mice can end up being used in animal studies, and only around 20% of early phase clinical trial participants are female. This has also led to what's known as the male default, the convention for findings from research which is conducted primarily on males to then be generalized to the whole population. And we find that even when studies do include participants of more than one sex or gender, it's not the norm for data to be disaggregated along those lines during analysis or when findings are reported. Fundamentally, research which doesn't account for sex and gender is less scientifically rigorous, and that it's less reproducible, and it's ultimately of poorer quality and use or usefulness. This creates and perpetuates gaps in the evidence base, which translate into compromised patient safety, less effective care, and worse outcomes for all people, but particularly cis women and girls, as well as trans, non-binary, and intersex people who are almost completely invisible in the medical evidence base. As I'm sure many of you will know in this room, women are almost twice as likely as men to develop dementia, and dementia is the leading cause of death in women. One reason for this is that women live longer than men, but it's not the only reason. And it's likely that there are different sets of risk factors for men and women. I hope I'm not preaching to the converted here. Some of those risk factors are going to be based on sex, i.e. biological factors, this would include gene variants such as the ApoE gene variant, which was being talked about earlier today, which seem to have more an effect on dementia risk in women than in men. Other areas which are not yet fully understood include the role of estrogen in protecting brain health. What I'm speaking to here is the fact that there are gaps in knowledge around how sex might be influencing Alzheimer's disease risk, and filling those knowledge gaps is a really key step for learning which medical treatments are going to be more effective. Other risk factors could also be shaped by gender, or what I mean by that being how a person's experience is shaped by the social world that they live in. One example of this might be that women have historically had fewer opportunities to go to university or to work in high-skilled occupations, and this may be having an important effect on dementia risk, but again, the knowledge is quite limited so far. As I'm sure some of you will know as well, there is this extraordinary statistic that 1 in 2 people will be affected by dementia in their life, either through having the disease or through caring for someone with the disease. And we know that women tend to make up 70% of unpaid carers, or up to 70%, necessitating there being not only a disproportionate burden on women, but actually I think that indicates there's a real need for kind of better sex and gender disaggregation of analysis in the kind of care and dementia care space as well. Despite this, despite all of these statistics I've just given you, male cells, animals, and people do continue, on average, to make up the majority of participants in dementia and Alzheimer's disease research. Moreover, sex and gender disaggregated analyses are rare in this field, and disaggregated data, which are critical for conducting really important meta-analyses, are rarely published in research papers. So as a result, women with dementia have ended up becoming what's known as a marginalized majority. So what are we doing about this? Well, there are long-standing policies to address sex and gender gaps in research kind of across the world, primarily implemented so far in Canada, the US, and by Horizon Europe. By contrast, in the UK, when the work on the Message project began in 2021, none of the major funding or regulatory bodies in the UK had a sex and gender policy in place. The MRC did launch a policy in July of 2023, but there remained at that time no unified sector-wide guidance for UK funders and researchers. So that's why the Message project was created. We've been working with stakeholders across the research sector to co-design a framework for a sex and gender policy for research funders to adopt. Adoption of policies based on this framework will mean that any researcher applying for funding will need to start saying in their application how they're going to account for sex and gender dimensions. We've co-designed this framework over the last 12 months with over 50 stakeholders, including research funders, regulators, publishers, researchers, patients, and people with lived experience, as well as clinicians, to ensure that it's tailored to the needs of users the whole way across the research pipeline. You can see here some of the organizations that have been involved in this work. Excuse me. And it's a very large and varied group. And I think the size of this group and the variety of it really speaks to a recognition that there's a need for a step change in this space. Alzheimer's Society are a big supporter of our work and have been very closely involved in designing the policy framework, as have Alzheimer's Research UK. Alongside the Association of Medical Research Charities and many of its members, as well as the NIHR and the MRC, who are going to be rolling out similar policy stipulations in coming months. Publisher stakeholders involved in the project include The Lancet, the BMJ, and PLOS Medicine. We've had researchers from a range of disciplines involved, both bench and clinical scientists, including researchers from the Women's Brain Project, which is a group that are pioneering research into sex and gender differences in brain health. So I'd recommend taking a look at them for more info. New expectations around sex and gender, as I say, are going to be rolled out in funding applications from as early as this summer by the Alzheimer's Society, and are likely to be embedded across the board over the course of the next year. We're also hearing from academic publishers that there's a lot of enthusiasm for increasing attention to sex and gender components in research papers, with a view to beginning to turn away papers that don't meet acceptable standards over coming years. Having said that, there's very much a recognition that there needs to be a phased approach to this, and the focus from all of the stakeholders that we're engaging with is providing guidance to help researchers to upskill over time. I'm just going to, oh, sorry, that's a little bit small. I'm just going to briefly run through some of the key areas of the message policy framework to give you some ideas to how to start thinking about this in your own work. This slide, which you can't see in huge detail, but it gives some details on the definitions of sex and gender, which have been designed to encourage precision in thinking around these terms, sex and gender. The focus here is really to draw attention to the specific sex and or gender mechanism that may be relevant to your study's research question. Previously, and usually, the terms sex and gender are used as quite broad brush labels, but we know that those terms each encompass a number of different features or characteristics. Sex characteristics listed here include genes, hormones, and anatomical features. And the message policy is really encouraging you to think about which sex and gender, sex characteristic or characteristics is going to be most likely to affect the medical condition or intervention that you're studying, and then to recruit participants to reflect that specific characteristic. Likewise, we ask researchers to consider the gender characteristic or characteristics that might be most relevant to their research question. For example, is it more important to know about a person's gender identity, so how they identify, their perceived gender, so how society or indeed the healthcare system perceives their gender expression, how they're outwardly presenting themselves, or their gender modality, whether they are cis or trans. Across all of these, researchers aren't expected to account for every characteristic. It's really about selecting the one which is most appropriate for your research question. And researchers conducting studies with human participants will need to consider if it's more important to collect data on sex, gender, or both to meet their aims. It sets out 4 key things to consider, describe, and justify in order to account for sex and gender. This is really quite granular, the granular detail of what it means to actually do this well. So the first thing is to say which sex and or gender characteristic or characteristics are you going to study, are you going to account for? Secondly, what's the target distribution that you're going to use in terms of sex and gender? And that could be a distribution based on the general population of society, a distribution based on your disease population, or a distribution based on a subset of the disease population. And very much the agency is with the researchers to decide which is best there. Thirdly, to give some indication of strategies for in clinical research, recruiting and retaining participants to meet that planned sex and gender distribution. In terms of preclinical research, giving some strategies for procuring and housing research subjects that are going to meet that distribution. In terms of secondary data, the expectation is that you will give a description of the sex and gender distribution of the original data set that you are using. Lastly, the fourth point here is giving details of any planned sex or gender disaggregated analyses, or if those aren't planned to be conducted, a justification for why not. Across the board, if the study will not account for sex and gender to meet any of these 4 criteria, researchers will be expected to give a strong evidence-based justification for this. And the policy itself includes details about which justifications may be acceptable or unacceptable. As I mentioned at the beginning, this policy is clear that researchers are not expected to have sample sizes that are powered to produce statistically significant results for each sex or gender group. It's more about thinking about how you can design your study to enable these factors to emerge in data at every stage of the research cycle. Our stakeholder group, which, as I said, includes a lot of funders, have been really clear that funders won't be viewing costs associated with better accounting of sex and gender as additional costs, but rather evaluating studies in terms of whether they are good value for money or not. I think that's really important, and it's a bit of a mindset shift. If a study is excluding 50% of the population, is it good science, and is it therefore good value for money? That's the kind of critical consideration here. And lastly, the policy framework makes a recommendation that researchers consider the sex and gender split of PPIE groups to ensure that the voices and expertise from those groups are representative of the affected population being studied. Just lastly, Cherie mentioned that guidance is absolutely at the core of what we're doing, and we're currently in the process of putting together comprehensive guidance to show you how to translate this policy into practice and into reality. The first thing we're doing is we're putting together 3 handbooks which really kind of walk you through each step of the process and how to actually do this well. There'll be one for preclinical researchers, one for clinical researchers, and one for funding organizations in terms of how to actually implement the policy. We have a website, messageproject.co.uk. On this website, in the coming months, we're going to be launching a database that we've been putting together, which contains best practice research in terms of sex and gender for a range of disease areas, including there's a sort of neuro area. If you'd like to have your research hosted in that section, please do let us know. We're constantly kind of welcoming new evidence to host in that database. And lastly, we're going to be running a short course through Imperial College, which is the university that the George Institute is affiliated with, on kind of new skills around sex and gender. And this is hopefully going to be launched early next year. So that's my bit over. I'm now going to hand over to Jess, who's more of a scientist than I am, to talk a bit more in detail.
My name is Jess, so I'm a research fellow in data science and epidemiology at UCL, and I'm also honorary fellow at the George Institute, where I did my PhD solely trying to understand the sex and gender differences in dementia. So today I'll present some of the studies I have done throughout my PhD and also a little bit in my postdoc. Here's just a talk overview. I'll go through some of the sex and gender differences that we look at in epidemiology and also traditional risk factors, including cardiovascular risk factors, sex-specific factors, and looking into medications. So here is a roadmap done by some of our colleagues, Sona Peters and Mark Woodward. This is trying to sort of go through the steps of conducting sex and gender desegregated in medical research. So the first phase is trying to explore the sex differences in dementia risk, presentation, diagnosis, treatment, and outcomes. And the phase two, we always strive to explain the sex differences by revealing some of the underlying mechanisms and ways to explain them. And then the phase three is to translate these differences that we found into interventions and policy programs. That's why the message program is extremely important in the last phase of this entire roadmap in sex and gender research. So first I would like to present one of the studies that we conducted, which is looking at sex difference in dementia risk and also risk factors. This was done by pooling 21 cohorts across 6 continents in 18 countries from the COSMIC Consortium. So what we found was the risk in the pooled data set was the risk of dementia, and in this case it's all-cause dementia, was higher in women in comparison to men. And we sort of look at this across each individual cohort, and we can see that it actually ranges quite differently. In some of the cohorts, the risk is higher in women, and in other cohorts, the risk was shown to be higher in men. And we, well, as I mentioned, all those cohorts coming from different geographical regions and have very different socioeconomic underlying background and ethnic background. And what we did was to look at some of the subgroups. So first, we found that across the different country level economies, from low to lower middle income, upper middle income, and high income, the risk of all-cause dementia was indeed higher, but this is most pronounced in the low to lower middle economy income groups. And when we disaggregate by birth year, those that were born before 1925, so the earlier birth cohorts, also show that the risk of dementia, which is higher in women, is most pronounced in the earlier birth cohorts, whereas the risk look more or less more similar in the later birth cohorts. So the hypothesis here is that, gender equity might be increasing, so the secular changes in improved educational and occupational opportunities over time in certain geographical locations may actually improve the disease risk that we see between men and women here. And from the same study, we also look at the sex difference in risk factor association with dementia. So it is really commonly assumed that risk factors work the same for women and men. So in this study, we want to investigate whether the risk factors indeed work the same or different for men and women in relation to dementia risk. Yeah, so on the left-hand side, these are some of the risk factors identified from the Lancet Commission report. And what we saw, actually, the risk factors do look quite similar in this pooled data set that we used, although there was some statistical significance showing that education might be more protective for men than women, and then we were able to quantify that using this metric called women to men ratio of the hazard ratios. And we look at, we use a different cohort, which is the UK Biobank, including over 500,000 participants, and we look at the sex difference in the risk factor associations for cardiovascular disease risk factors and also incident all-cause dementia. So similarly, we look at some of the major cardiovascular risk factors in relation to all-cause dementia risk. And what we can see, if you sort of focus on the blood pressure section, you can see that the direction of association for women and men, they're sort of opposing. They look quite different. With higher blood pressure, sort of have this dose-response relationship for women, but this is not really the case for men. The risk seems to be decreasing as the blood pressure increases. And if you also look at BMI categories, there is also some sex difference in terms of in the overweight and obese group in relation to healthy weight. We can see the risk increases for women, but this is not as of a strong case for men. So we decided to visualize this using splines. So this is a good way to sort of look at the continuous association between the variables of interest and the risk factor of interest and the hazard ratio. So the first panel is systolic blood pressure, and then middle diastolic blood pressure, and last panel is BMI. You can see within the systolic blood pressure panel, the association seems to be U-shaped for men, which is the blue one, blue and purple. And the red one showed a dose-response relationship, and that is for women. So this is quite clearly distinct because you can see the separation even in the 95% confidence interval. And what we proposed here in terms of some hypotheses is more related to biological. So there has been evidence around sex difference in orthostatic hypotension, so the risk of low blood pressure, which can induce head and brain injuries, and this is more prominent in men. And head and brain injuries have been shown to be a very strong risk factor for dementia as well. Unfortunately, we didn't have enough data here to sort of look a bit deeper into potentially the mediating effect of brain injury. And possibly the separation in BMI indicating different body types and between men and women, and possibly a different BMI cutoffs are needed for women and men in assessing dementia risk. So apart from trying to understand the sex and gender differences in risk factors, another way when we see a higher risk in one sex than the other, the contribution could be stemming from sex-specific factors, which means the factors that are restricted only pertained to one sex or gender only. So in this study, we look at the reproductive factors, which are more related to women and the risk of incident dementia using the UK Biobank study once again. So we look at various reproductive factors across the entire reproductive lifespan. So from early life, such as age and menarche, the first period, and also the use of oral contraceptives, to pregnancy, first birth, reproductive years, all the way to menopause. So what we saw was that the cumulative estrogen level, whether it's endogenous or exogenous, if the reproductive events indicates a lower cumulative level, that is associated with a greater risk of all-cause dementia for women. And one thing I want to draw your attention to is the number of live births. And this is on the right-hand side in the red box. This is for women. So we looked at this for women that had 0 live birth, 1, 2, 3, 4 more. We sort of see like a U-shaped relationship with 2 as the reference. in terms of dementia risk. But we also look at this number of children in both women and men. So number of children fathered in men was the sort of the comparator here, and we saw a similar sort of U-shaped relationship between men and women in terms of number of children and the risk of dementia. So what we can deduct from this observation is that possibly the the number of children and the risk of dementia for women, we see this U-shaped relationship. Maybe it's unrelated to biological factors which involved in childbearing, which only a woman is able to carry a pregnancy through. But we saw the similarity for men as well, which indicating this is possibly due to socioeconomic factors involved in the parenthood for both women and men, rather than the childbearing, the biological, physiological changes that increase the dementia risk. Okay, so now I'm going to move into medications. So we looked at two trials. So the PROGRESS trial was blood pressure lowering treatments with the primary outcome to kind of look at recurrent stroke in people that already had a stroke or transient ischemic attack. So we decided to look at this in relation to cognitive decline or dementia as a composite outcome here. And just to investigate whether, blood pressure lowering treatments in preventing subsequent cognitive decline and dementia may be different for women and men. And what we saw is that there's actually no difference, which sometimes is a good news, that both women and men should be treated equally for their blood pressure lowering. And then we look at a different trial, which is was the ADVANCE trial. So this is in diabetes patients, and the interventions were a combination of glucose lowering and blood pressure lowering treatments. And we look at the same thing in relation to cognitive decline and dementia as the outcome for this randomized therapies. Now, once again, we didn't actually see any sex difference, which, it's indicating that women and men with diabetes should be treated the same in terms of their glucose and blood pressure lowering treatment regime. However, when we move into some of the more recently approved anti-amyloid drugs, me and a colleague in Harvard and also Mark from the George Institute, we decided to write this commentary, which was published on JAMA Neurology, which is a piece to call for action for those big anti-amyloid drug trials to report sex differences in treatment effect. So what we saw was that the drug lecanemab, if you're sort of looking at the red box across all these plots, you can see that the drug seems to only work for male, but for female, the confidence interval actually crossed unity, which indicating there was no effect on some of the improving some of the cognitive measures. I don't think we have heard back from some of these trials and whether they have done further analyses or reanalyses, but I guess one important thing to note was that all these sex difference findings were only in the supplementary. It was not included in the main text. It was not included in the headline findings, even though we can see there's a clear sort of sex difference indicated by very small, potentially very small overlapping of their 95% confidence intervals. So another thing about trying to understand I guess, women, sex and gender representation in clinical trial is to look at are women and men fairly represented in some of the recent dementia trials. So we conducted this systematic review, and what we found was a total of 118 trials, and out of this eligible trials, 113 reported sex of the participants and were included, and hence it was included in our analyses. And of the 110,000 participants in these 113 trials, 58% were women, which indicated a representation of women and men. However, the global prevalence of dementia population is around 64% women, which indicating that it is still a lower representation of the clinical trial populations in comparison to the real underlying population how many people that actually have dementia in the real world. So, but interestingly, women's participation in these trials tend to be higher when the first or the last author of the trial reported were women. And 8 out of the 118 trials reported sex disaggregated outcomes, and only 3 of those found significant sex difference in efficacy outcomes. So overall, women and men were equally represented in dementia trials carried out over the past decade, but women's representation was lower than the actual underlying dementia population. So sex disaggregated efficacy and safety outcomes were very rarely reported. So thanks for listening, and I will conclude my presentation here.
Thanks, Jess. I just have one final slide which I wanted to present, which is a bit of a call to action to those of you in the room. Basically, when you are reading about research or even conducting your own research, have a think about these 5 questions. How many female and male participants are there? Are results reported separately for females and males? Are the data disaggregated by sex and gender in the kind of analysis that's being conducted? Is there any attempt at all to go beyond the binary into the to kind of account for people trans, non-binary, and intersex folk? And lastly, do authors reflect on sex and gender in the discussion? And that's really drawing out some of these key learnings that we need to start generating in order to start to improve knowledge in these spaces. So yes, we are now done. Thank you so much.